Morning. It's now my pleasure to introduce our next presenter, Dr. Christian Grøndahl, a co-founder and CEO of Microbiome. Again, the format for the session is a presentation followed by some Q&A. If you'd like to ask a question, please type it into the box underneath the player. Thank you for joining us today. Please begin with your presentation.
Thank you, Gudo. And thank you, Sharon, for giving us the opportunity to present Microbiome here today. So Microbiome is a CRISPR technology and also gene therapy company. And our vision is actually to become one of the leading CRISPR and microbiome companies globally. So our CRISPR platform has therapeutic utility, both in selectively killing of bacteria, but also in reprogramming of the microbiome in terms of gene therapy of the microbiome. So up to now, we have been focusing our microbiome modulation in diseases with very high unmet medical need. So our leading program is within infectious diseases and antimicrobial resistance. We've also focused a lot on our CRISPR platform, and then we have some early projects within cancer and gene therapy of the microbiome. We raised a very large Series A, actually the largest Series A here in Scandinavia back in 2019 from four very strong investors. So we raised $50 million from the Lundbeck Foundation Emerge, from Northeast Family Office, from Life Science Partners, and from Wellington Partners. And we were the first company globally to be awarded a broad US patent back in 2017 for CRISPR precision bacterial killing in microbiomes. So over the years, we've grown, and we now comprise more than 50 scientists, mainly PhDs, with backgrounds in academia, biotech, and pharma. And this is sort of a timeline of our joint history and joint achievements. So we were, as a company, inaugurated in Cambridge, UK, back in 2015. That was also where we made some of the seminal studies and inventions around CRISPR and microbiomes, and we submitted our first patent application. Then we managed to get a seed funding in 2017 from the Lundbeck Foundation Emerge, and we actually moved the company to Denmark, and it was in conjunction with having our first US patent awarded. We then, as I said, secured this large Series A in 2019, and we then used the funds to actually make our first clinical development candidate in 2020. And this candidate, we are then fast forwarding into the clinic, and we have now an open IND with the FDA that was opened in December of last year. And just in January of this year, we actually obtained the fast track status from the FDA. Along the way, we have been able to go into three very valuable partnerships. So we have a collaboration with CARB-X that are sponsoring our first clinical program. They've also been supporting our preclinical activities with a donation of up to nearly $10 million. We also have a research and development collaboration with one of the leading cancer centers in the world, MD Anderson in Texas and Jennifer Vargo. And this is in an area of immune oncology and trying to improve the treatment of combined checkpoint inhibitors. That is on an undisclosed target. And finally, we have a very exciting collaboration with Novo Nordisk in the area of gene therapy of the microbiome. And this is in the field of cardiometabolism and NASH and diabetes. We're very fortunate to have a very strong leadership here in the company. Milan is leading our R&D as our Chief Medical Officer, and his large team is actually very experienced. So together, we have more than 120 years of experience in drug development. We are three founders of Microbiome, and here's my two co-founders, Professor Morten Sommer from the Danish Technical University of Copenhagen, and also patent lawyer Jasper Klub from Cambridge, UK. Likewise, we're very fortunate to have a very strong scientific advisory board with these eminent scientists. Our advisory board is chaired by George Church, professor at Howard and MIT, and a true pioneer in synthetic biology and CRISPR applications. Peter from UCSF and Harris from Columbia are both experts in the human gut microbiome and in genome engineering of the human microbiome. Joe is an eminent CRISPR and anti-CRISPR expert, and Federico is a very experienced gene therapist. So now I'd like to turn to our CRISPR technology and the various therapeutic applications that we are using. So the logic in this slide goes from left to right. So in all applications, it's very important for us to have the delivery of our CRISPR-guided vector, with we dubbed a CGV. And when that enters into the bacteria, we can have several modes of actions. Either we have the CRISPR RNA and Cas complex to seek out the target genes to making a gene destruction, and that actually leads to very fast and efficacious bacterial killing. That is the mode of action for the two top swim lanes, the blue and the green. But we can also use the CRISPR-Cas complex to introduce a gene, and then we have a gene therapy of the microbiome when we have the production of a therapeutic protein or peptides, and that's the yellow swim line. The various indications that we are using in the top blue swim line, that is in treating intractable infections, typically with antimicrobial resistant bacteria. And here our lead project is trying to prevent bloodstream infections in very vulnerable cancer patients with multi-drug resistant E. coli. We also have a sister project where it's actually treatment of the bloodstream infection by IV administration of our CRISPR medicine. And then finally, we also have an undisclosed target of a dysbiosis indication. In the green swim lane, we are targeting, again, bacteria for selective killing, but these are no longer infectious pathogens. These are bacteria that are associated with disease. And here we are working both in immuno-oncology and also in colorectal cancer. And in the yellow swim lane, where we are applying the CRISPR technology for having gene therapy of the microbiome through a disease intervention with the synthesis of a therapeutic protein or peptide, this is where we have the collaboration with Novo Nordisk in the cardiometabolic space on an undisclosed target. And we also have our own gene therapy of the microbiome project in an indication of IBD. So this is another way of looking at our pipeline. You will see the blue swim lane, infectious diseases, the green, the disease-modifying bacteria, and then the yellow, the gene therapy of the microbiome. Our most advanced project is just on the edge of entering into first human dose trials later and next quarter. And this is where we have the collaboration with CABX. And later in the presentation, I will give you, more details about our most advanced program. We have the sister project where we are treating multi-drug resistant E. coli, and here we are planning to make our first development candidate later this year. In the green swim lane, we are targeting pathogens that are associated with the pathogenesis of colorectal cancer, and also this rare genetic cancer, familial adenomatous polyposis, or FAP. And in that area, we also plan to have a development candidate to be identified and profiled in this year. Then we have an early collaboration, as I said, with MD Anderson, and here we are targeting a modulation of the microbiome. So we hope to see a much less toxicity when these patients are receiving combined checkpoint inhibitor treatment. And then in the gene therapy of the microbiome, we are progressing a project together with Novo Nordisk in the NASH, obesity, and diabetes field. So now I'd like to shift gears a little bit and take you deeper into the data that we have generated supporting our IND submission and the data that we have compiled around our first development candidate asset, SNIPR 001. When designing such a CRISPR drug, we have two orthogonal delivery mechanisms that we can choose from. It can either be that we design and CRISPR arm live bacterial phages, viruses that prey on a bacteria, and then have the phages to inject the CRISPR-guided vector directly into the bacteria. So that's what we call a CAB or CRISPR-armed phage. An orthogonal delivery system is actually where we're placing the CRISPR kill circuit or the CRISPR information inside a donor bacteria, and then these donor bacteria then meets the target bacteria, and then via a horizontal gene transfer or conjugation, we have the CGV to be delivered to the bacteria. Regardless of the delivery route, we have the same, synthetic biology and processes happening inside of the bacteria. We have the CGV DNA to be transcribed into a CRISPR RNA, and also a Cas protein, a Cas endonuclease, that is actually the protein that are able to make double-stranded breaks in the DNA. When the CRISPR RNA and the Cas makes a complex, they seek out the genes that are fitting the CRISPR information, and then they make a cut, and that leads to the bacterial cell death. We have a focus here at SNIPR Biome on trying to tackle the growing health problem with antimicrobial resistance. And this is actually a very big problem. You would think that there are many more biotech companies that would be, targeting the growing health threat with antimicrobial resistance. There was just a Lancet paper some weeks back that really described the global burden of bacterial antimicrobial resistance, and it's a staggering 3 to 4 million each year that are actually dying from this, a slow tsunami that are simply just getting more and more traction. And that, you could say, is as big a problem as the ongoing pandemic with COVID. We have seen that the most important pathogen here in the list is actually E. coli, and that's one of the reasons we are focused here. But there are others that are urgent threats or serious threats. It could be C. difficile, it could be Acinetobacter, it could be Pseudomonas, it could be Staphylococcus aureus. We actually believe that our CRISPR technology has the option to make effective drugs tackling this antimicrobial resistant crisis. And we are very eager to bring the first drug into man targeting multi-drug resistant E. coli. We are very fortunate and proud to have this sponsorship from CARB-X, and they have been sponsoring us through the preclinical stages, and now they're also sponsoring a part of our clinical activities, and that is actually a very substantial support and validation of our efforts. Also, we've been very happy to see the very appreciative feedback from the FDA, opening of the IND late last year, and then obtaining this fast track status, which of course also is a validation of the big unmet medical need and the data that we've been able to provide. So here is now a further look into how does our first drug candidate look like. And you will see here in the cartoon that that our drug is actually a living drug. It is comprised of 4 different bacteriophages. They are all living, and we have CRISPR armed them with our various CRISPR kill circuits. And we've also changed the tail fibers. I have here a small 3D print of a phage, and it's actually these tail fibers here where they land on the bacteria that one can actually moderate and change in order to get a much broader host range. And that's actually what we are trying to obtain here with this cocktail of 4 bacteriophages. We actually want to go into as broad a collection of the E. coli biology as possible. Again, when the DNA enters into the bacteria, the E. coli specific CRISPR RNA will be transcribed, and the Cas nuclease will be active. Together they form a complex. They find out the target genes, and they make a double-stranded break, which leads to cell death. This is the target product profile of our first CRISPR drug, and you will see that we are actually targeting to have a preventative treatment, to have a prevention of bloodstream E. coli infections by eradicating the E. coli in the gut before they translocate into the blood of the patient. So the lead indication is a prophylaxis of bloodstream E. coli infection in patients with hematological malignancies. And we have chosen these patients because they have, both due to their disease, but also due to their treatment, they have a very low ability to fight off an infection. Their own immune system is hardly existing, and once they get a bloodstream infection, they have a very high risk of dying. We have brought some very exciting preclinical data, and I brought some of the data here, that in a model, in a murine model of human disease, where we have colonized these mice with human pathogenic E. coli, we're able to see a clinically meaningful and very substantial reduction of E. coli in the gut of these mice, from around 10 to the 8 or 9 down to 10 to the 4th. So between 4 and 5 log reduction, or 10,000-fold reduction of the bacterial load. So we think that is very clinically meaningful, and we hope to be able to see also substantial decreases in the clinic. So the stage of development is that we have an open IND. We might actually be eligible for following this fast track. It's called LPath, limited population pathway. And with the fast track designation we got from the FDA, we will look forward to go into first human dose here later in this first half of this year. Very importantly, we have shown a very high coverage across all the clinical strains, all the pathogens of E. coli. So what I'm showing you here is sort of the phylogenetic tree of a very large collection of E. coli that has been isolated from the blood of specifically these cancer patients that have been suffering from a bacterial infection in their blood. And you can see that these bacteria can both be grouped into phylogroups and also these multilocus sequence types. But regardless of the nomenclature, you will appreciate in the outer green ring that we, with green, actually kill more than 90% of all of these pathogens. So we think that is actually very substantial. Equally important is that we seem to be able to have a very high coverage of the E. coli strains that are resistant to the known and used treatment. So fluoroquinolone is actually a treatment that is used prophylactically in these patients. It's not approved, but still it's actually used prophylactically because they have a high risk of bloodstream infections. But we're seeing now many E. coli strains becoming fluoroquinolone resistant, and you will see here that we are able to target more than 80% of these fluoroquinolone resistance. Likewise, for multidrug resistant, ESBL, and carbapenem resistant E. coli, we have a very strong coverage. And then equally important, we've also documented that we are very selective. We are only killing E. coli. So if you look at this panel here, we have documented that the 001 have no detectable off-target effects of some of the other constituents of the human microbiome. So we are very thrilled to bring this into the clinic and hope to be able to see equally good results in humans. So now I would like just to very briefly also mention that besides in infectious diseases, we are active in immune oncology, as I said, in the collaboration with the MD Anderson Cancer Center and Jennifer Vargo. We entered into this very exciting collaboration last year, and we look very much forward to see if we are able to change the microbiome to a phenotype that is then associated with less toxicity of these very exciting new treatments with immune checkpoint inhibitors. There's no doubt that there's emerging evidence of the importance of the microbiome in immune oncology outcomes, and we have now seen some very interesting dynamics. Some bacteria are very wanted, other bacteria are very unwanted, and we will try to modulate the microbiome to see much less toxicity. We're also active in cancer, and this is an early project, but where we're trying to really address some very exciting biology where some bacteria are associated with the progression and the pathogenesis of colorectal cancer, and also this rare genetic cancer, familial adenomatous polyposis. And here we're trying to target specifically the bacteria that are producing a toxin that happens to damage the DNA of human cells. And we really are very excited about this prospect, because we actually think our CRISPR technology are excellently suited to take out just a subset of bacteria where these genes that are producing the toxin can then be targeted. And then finally, to the third application of our CRISPR technology, the gene therapy of the microbiome. And you could argue this is where we have the biggest transformatory potential. What if we could actually design CRISPR medicines that are then delivering a gene to the microbiome, and then the therapeutic of choice will then be made by the microbiome personally to the patient that needs it. This is where we have the collaboration with Novo Nordisk. And again, we have to decide what is the best delivery. Is it by making CRISPR-armed phages, or is it actually by having the CRISPR information in a conjugative bacteria? But in contrast to the other applications, here we're not aiming to disrupt the genes and making a selective bacterial killing. Here we are actually aiming to insert the gene and then have a therapeutic protein or peptide expressed into the gut microbiome. We are very excited about this collaboration with Novo Nordisk. It is on an undisclosed target, so I cannot go further into that. Other to say that we believe it's very obvious to work with one of the pharma giants in the cardiometabolic space. But there are a very big potential for making these in situ productions. One could think of having production and secretion of agonists. It could be gut hormones like the GLP-1 or GLP-2. It could also be antagonists like full-length antibodies or domain antibodies or fragments, or it could be the productions of enzymes to try to modulate and direct a metabolism in the gut microbiome. I believe that the applications here are very powerful, and there are plenty of biologies and targets to go after. So just to finish off, what we are doing ourselves is that we want to make a platform where we can have in situ production of nanobodies using the SNIPR technology, and these nanobodies could then neutralize toxins. They could be active in immuno-oncology. They could be used in chronic immune settings. The list is long. But the way we do it is actually we clone CRISPR into these vectors, have them to be delivered into the resident microbiota. So this is not where we're actually giving a bacteria that needs to colonize and be a member of the microbiome. Here, our drug is a DNA that then moves into the resident microbiota and then gets an expression from there. And then I'd like to finish off my presentation by saying that this very strong CRISPR technology is also parallel with a very strong and growing CRISPR and microbiome patent portfolio. So we have an early and broad position in the US. We were the first company to get a patent there in 2017. That estate has grown to 20 granted US patents. We've also been the first to get a broad patent in Europe. Basically, we have coverage of our CRISPR technology to use whatever Cas, Cas3, Cas9, Cas12, whether the Cas is exogenous or endogenous, and we can target whatever bacteria in whatever indication. So we actually really hope to be seen not only as innovators in this CRISPR modulation space, but also to be an enabler and to forge many partnerships or licensing deals. And I really invite you to contact us if you have a project that you would like to collaborate on. And with that, I'd like to thank you for your attention. And then over to you, Gula, for questions.
Perfect. Thanks. Super interesting. I think we have time for a couple of questions. And I think I'd like to just ask, maybe you can elaborate a little bit on what makes the CRISPR-armed phage approach different from, let's say, other approaches that aim to use phages as something like narrow-spectrum antimicrobials.
Yeah, no, that's an excellent question. And, phages are really efficient bacterial killers. And of course, there has been an arms race between, bacteria and phages for, millions, if not billions of years. But typically, phages are very narrow spectrum. So if you have, a phage, it will be very, very picky and only go into a very narrow sliver of, for instance, the wide E. coli biology. So we have had a task to find not only native phages, but also phages we could modulate, so we could broaden the host range, and with 4 phages actually cover the whole E. coli biology. That you cannot do with a natural phage. Then, moreover, we have armed the phages with CRISPR, and that means that we have, very specific ways of disrupting the DNA of the bacteria, and we are not relying on the entire phage cycle to have the lytic effect of the phage. These phages are lytic, and that's actually very clever because they will then also propagate and divide where there is an infection. But our CRISPR-armed phages will actually also kill a bacteria that is not dividing, and that is something that a natural phage cannot do. They are relying on that they live together with the bacteria, and the bacteria has many ways of invading a phage. So we think that bacteria that are hibernating or in a microfilm or is actually dormant, we can also kill them with our CRISPR-armed phages, and we have data to support that.
Fantastic. So I think we're at the end of this session. So I'd like to thank you again for your participation and for the great overview of SNIPR Biome.
Thank you, Gula. Thank you for the opportunity to present SNIPR Biome.